My lab, like so many of the others that have talked already at this meeting, is interested in going around this circle that we've seen so many times. We're interested in taking patient cells, reprogramming them to iPS cells so that we can direct their differentiation to clinically relevant cell types that would allow us to do disease modeling in a dish, or in some cases transplantation studies, or we want to use those cells as a substrate for drug or genetic screens, and we're hoping that all of that will feed back to patients. Now, in an ideal world, we'd be able to simply order some chemicals that we could just add to our dishes and get 100% of our cells to differentiate to be reprogrammed from patient cells to iPS cells. And then we could order other chemicals that would allow the directed differentiation of those cells. Or you could imagine even just ordering one tube that would get you straight there. But right now, we don't have that capability. And so what we're currently doing is using gene transfer and transgenics both as a way to reprogram our patient cells into iPS cells and as a way to purify those cells once we do the directed differentiation, since it's never perfect. So today I'm going to segment my talk into two parts. I'm going to talk initially about reprogramming. And in the second part of my talk, I'm going to talk about using genetic markers to be able to purify those cells after differentiation. So I'm not going to spend too much time talking about this, since I think everybody in this audience knows a lot about it already. Suffice it to say that we use, still in my group, what I'm going to call Yamanaka's Fantastic Four, or the Fantastic Four, to reprogram patient somatic cells to iPS cells. And in the very beginning of this work in my lab, there was a three-way collaboration between Michel Sadelain's group, Lorenz Duder's group, and my group. And that initial work relied on the use of retroviral vectors that can be called diploid. These are positive-stranded viral vectors. One of the reasons that we use them is that gene transfer is very efficient with these viral vectors. Once that gene transfer has occurred, these positive-stranded templates are reverse transcribed and turned into double-stranded DNA that is then integrated into a host cell's genome. And the function of that integration means that you get sustained, strong transgene expression for the length of time that it's going to need for reprogramming to occur. So in this case, you get strong expression of the Fantastic Four, and then a reprogramming event occurs. Irini Papapetrou was a postdoc that was in Michel Sadelain's group, and she's the one that brought the system to my lab initially. And she now heads her own lab at the Washington School of Medicine in Seattle. And Irini designed a series of vectors that have spectrally distinct fluorescent proteins that are then coupled to the Fantastic Four. And so it's kind of a cool system because it allows you to see the expression of each of these reprogramming factors in individual cells. And it gives you the granularity to be able to go back and sort out different populations and see what happens during reprogramming. But even though that method and those cells that we made from that original study have been very useful, we knew at that time that there were some issues with them. And of course, the obvious issue that everybody knew about was that you can have insertional mutagenesis occur with this type of technology. And these transgenes don't always silence. So it's useful to have them around early to get the reprogramming to occur, but later they're sort of a hindrance. One of the things that wasn't so clear is that these transgenes can reactivate during differentiation. We always worried about that. But one of the things that wasn't obvious is that also over passage, you can get reactivation of these transgenes. And that was something that we hadn't anticipated, but was revealed by the fact that fluorescent proteins were marking the expression of these different reprogramming factors. So we were a little surprised and troubled to see that. But essentially, we knew from the beginning that we wanted to move to transgene-free reprogramming. And of course, there was, around that time, an explosion of different techniques and technologies that allowed transgene-free reprogramming. But from my own personal perspective, I wanted to focus on something that used RNA and not DNA. Because in my opinion, as soon as you involve DNA, you have the possibility of getting a mutagenesis event occurring. You can have accidental insertion of DNA, and it's hard to prove that negative. So that led me to focus really on the RNA-based methods. And there were really two main ones. There was Derek Rossi's modified RNA method, or even protein-based transduction, which might be the most direct. And there was a Sendai virus-based method. And when I thought about this, you know, this is a variation on Waddington's landscape here. When you think about trying to reprogram cells, it's an uphill battle, right? You have to have, you're starting down here from a somatic cell, and you want to push the cell up to the top. And what that means is that you have to continually transfect RNA in if you're going to use the ROTC method. And so for that reason, I really thought that the Sendai virus method was going to be the way to go. And so I was lucky enough to contact Dr. Noemi Fusaki, who's at this company, Dynavec, very early on, and she's been a wonderful collaborator and been very helpful to us in helping get this technology into my lab. The way that this vector works is that it has a negative stranded RNA genome, and I should just point out, of course, because it's a viral vector, it's also very efficient at getting gene transfer to occur. Once that negative stranded RNA genome comes into the cell, it can be used to create positive stranded RNA that can be used to make more viral proteins and Yamanaka's Fantastic Four, as well as can be used as a template for making more negative stranded RNA genomes, okay, to propagate itself. And the beauty of the system is that the virus does all of this in the cytoplasm, and there's no DNA-based intermediate, so you don't have to worry as much about integration into the host cell's genome. The only issue that I've had, and I think the only thing that really stops the massive adoption of this technology worldwide, which may happen anyway, is the cost, and it's a little bit expensive. And so my group has been trying to figure out ways to be more thrifty with our use of this technology. And so the question is, can we use less of this kit to still reprogram efficiently? And so one of the experiments that would help, and one of the ideas was, could we sub-aliquot out each kit and then refreeze it into smaller aliquots that then could be used as we needed when different people had come into the lab. So we did an experiment using a Sendai viral vector that expressed GFP that again was provided by Dr. Fusaki. And I put one of these through a freeze thaw and left the other one fresh as a control, and did a dilution series, transduced cells, and then did FACS analysis to look at how much titer we lost by performing that freeze thaw. And here, before I did the FACS analysis, I just took some quick pictures after doing a multiplicity of 3 transduction and then a 2-fold serial dilution. And I think you'd probably agree with me if you just look at the cells, there's not a huge difference here, which was encouraging. And that was borne out by the flow. So here, if you look at fresh samples versus the freeze thawed, you can see small differences, like this peak out here is a little bit lower, but it's not a lot lower. And so if you run the numbers, you figure out that to equalize a freeze thawed sample, you can multiply the volume by 1.34, and that gets you roughly back to where you were before you did the freeze thaw. So it was encouraging that we were able to do a freeze thaw and not lose too much titer. And one way to get that transduction efficiency back might be to do things a little more efficiently. And so my colleague Sunita D'Souza at Mount Sinai, in a meeting that we're just starting to do called Coordinate, this is a group of core facilities that are getting together and trying to share ideas and technologies. Sunita suggested that I try transducing cells in suspension as opposed to being attached to the dish. And so I went back after our meeting to my lab and tried this experiment out. And the way that it was done is to take the same number of cells, but take them in a small volume of media, add the virus directly to that small volume, and leave it for an hour at 37 before then plating it in the same size vessel. Okay, and so that was the experiment that we did. And we found out that Sunita was right, that if you look here, especially at lower titers, lower numbers of viral particles, that there was a much more efficient transduction efficiency here. So I think that this could provide a way forward for allowing us to regain some of that lost ground by refreezing these aliquots. And then I just want to end on sort of a cautionary note, which is that in my hands, different patient fibroblasts are reprogrammed with very different efficiencies. So here you can see these were 2 different patient fibroblast lines where I actually took the skin punch biopsies, made the fibroblasts myself, transduced them side by side, so all of those variables were removed. And what you can see is one line is much, much better at reprogramming than the other. So the question that we have now is how far down can we go, how small can we go before you start to miss certain lines? And that's an answer I don't really have for you right now. We're going to have to try to work on that further. But what is clear is that this is a very routine, mature technology that I use all the time in the lab now. And as far as I'm concerned, the matter is pretty much settled. And so now I'm going to talk about trying to create genetic markers to purify cells, because once we've made those iPS cells, we need to try to study them by doing directed differentiation. And we want to get clean populations out at the other end, such as neural cells here. And so to be able to illustrate this, I want to turn to one of the projects that's going on in my lab. And what we're trying to do is make iPS cells from autistic children which is one of the most prevalent developmental disorders, and depending on whose numbers you want to believe, it's roughly about 1 out of every 100 children suffer from some disease in the autism spectrum. It's got a very strong genetic influence if you look at twin studies. And the problem has been for the field that you cannot obtain neural tissue from these patients, so it's been very difficult to study. And it's very difficult to model in mice. If you look here, this is a slide that I've adapted from Paul Patterson, who does try to study this sort of disorder in mice. It's obvious that mouse behavior is pretty subtle, right? So there's certainly a role for trying to understand this sort of behavior in mice and to try to model these things in mouse. But we wanted to take the opposite approach, which was to use the clinical data from certain patients and then make iPS cells from them and turn that neural. Okay, so that's what we've done here. I'm showing you that we've used the Sendai, the cytotune kit, to make a number of different iPS lines from these different patients. One of the things that happens with autism is that about 1/4 of the patients suffer from seizures, and it's not really known why. So I'm going to focus on that question here right now. And you can see here we've got a few of the patients that do suffer from seizures, as well as one that does not, as well as, of course, the apparently healthy controls. So this is obviously a vast oversimplification of how you could think of cortical activity, but in the most general sense, you can imagine that there are inhibitory GABAergic forebrain interneurons that are trying to tamp down the excitatory glutamatergic neurons, and that interaction between these two cells gives you a level of activity, of electrical activity. So in the case of these patients, it's likely that either there's a loss of inhibition or a gain of excitatory information. Okay, and so what we wanted to do was directly be able to test these different hypotheses. And to be able to do this sort of experiment, you need two things. You need a directed differentiation protocol, and then if it's not perfect, which it never is, you need a way to purify your cells. And I was lucky that I was able to cheat because Asif Maroof was a student that has worked for a very long time with me. Hopefully some of you saw his talk. And he was a graduate student that was cross-appointed between Stuart Anderson at Weill Cornell and Lorenz Studer. And these guys were nice enough to share their unpublished protocol with me. So I got a get-out-of-jail-free card. I was able to pass the differentiation and then focus instead on trying to prospectively isolate the desired cell type. And I was also lucky that at that time, I had already started working with zinc finger nucleases through a collaboration with Maria Jason and a very talented postdoc in her lab, Erica Brunet, who's since gone on to start her own lab in France. And what we were trying to do is engineer translocations using these zinc finger nucleases in a clinically relevant cell type. So I was providing them with ES-derived mesenchymal progenitors, and we were engineering zinc finger nuclease, or engineering translocations using these zinc finger nucleases. We published that in 2009, and you can see here a fish image showing you that we were able to get this done, doing this sort of thing. But at that time, I was also playing around with using these zinc finger nucleases for homologous recombination. And we knew that it worked very efficiently to be able to cause homologous recombination, force a homologous recombination to occur in iPS cells and DS cells, actually. So what we wanted to do was put in a reporter, in this case LHX6 GFP, that would identify these ventral forebrain interneurons. And we had a region of homology surrounding it. So this construct was created by Kathy Jubin in my lab. It was then given to Amelia Richardson, who used it as well as this zinc finger nuclease that would cut in this spot called AAVS1 that is thought to be a so-called safe harbor, a place that we can just put a gene specifically. Okay, so the hope was that instead of randomly integrating the transgene, we could place it here. So Amelia went to the lab, and she took H9 human yes cells, and then she nuclear affected the construct I just described along with the AAVS1 zinc finger nuclease into the cells. We selected and then expanded these different lines. And then she used Asif's protocol to differentiate them into putative forebrain interneurons. And we screened for green. And it worked. We got a number of different clones that had GFP expression when using this protocol. And then we designed a method that would allow us through PCR to determine clones that had undergone correct targeting versus those that had random integration. So we used a primer inside the gene as well as one outside of the region of homology. And in this case, we'd get a PCR product of a given size. Whereas if you had random integration, the PCR itself should fail because the primers would be too far away. And so here you're looking at one of the gels that Amelia made, and you can see the clone names that are in green are all the ones that express GFP. We included one that did not as an internal negative. And you can see the band of the correct size and a number of these different clones. And 3 of these lanes where it failed are all clone 16, and this is because Amelia is a very thorough individual, and she wanted to make sure that the lack of product was not something technical. And so you can occasionally get random integrations, but very frequently you get homologous targeting, which is exactly what Rudy Anish talked about in the very first session. And the problem that we've had is that this construct is dim after integration into this locus. So while it works, it's not very bright, and that makes it technically difficult to use to get sorted cells, for example. And in the meantime, what's sort of cropped up is this new technology called talents, which are essentially the same thing, except you can rationally design them in a computer, and that makes it much faster and much cheaper to use. And I think that this is really going to change the field. We were using AAVS1 because there was an off-the-shelf kit that we helped Sigma beta test, and so it was useful as a general place to try to put transgenes. But now, using this technology, we should be able to knock directly into the endogenous locus. And in my opinion, this is really going to be the way forward. I think that as a field, we're going to have to do this all the time, routinely. And again, I just told you that talents make it easy and cheap, and of course, as a small free plug to my hosts, I want to point out that they now have a commercial service that allows you to do this without having to think about it too much, which is nice. And so we can now knock in fluorescent proteins to identify any cell type of interest, or introduce or revert mutations. And of course, all of this has been shown already by Rudy Anish's lab, which I think underscores why he got the award that he did. And in the end, we need to do this so we can really verify that the phenotype is following the genotype in a very clean and directed way. So using isogenic cells, we want to make sure that this isn't just variation that we're looking at. We're going to conclude is due to the disease process. So just to summarize, in a couple of years now, we've been able to move from a situation where we had random integration through lenti reprogramming, as you would, as sort of in cartoon form shown here by these blue dots. And we've moved away from randomly integrating transgenes as a way to purify cells. And using the Sendai method, we can now have clean genomes, and then we can very precisely place where we want to put our markers. And I think that when you're comparing healthy to disease state cells, it's going to allow us to compare apples to apples instead of apples to oranges. And it should really reduce the amount of noise that we see in these sorts of assays. But there is a cautionary thought here, and you know, I don't want to be too pessimistic, but that's somewhat my nature. And you have to be concerned that the gene itself might not be expressed at a high level. So what I've told you is that this construct, when we integrate it into AAVS1, is expressed at a very low level transgenically. We know from Ossoff and Stuart Anderson's work that in transient situations where you have lots of copies of this construct around, that it is actually quite bright. But what we don't know is what's going to happen when we knock it in, and it's possible that we're going to see a situation like this. And so it's important to remember that when you're knocking in something like GFP, you're reading the transcription of a gene of interest, and the transcription of a gene of interest might be low. Okay, so that's something that has to just be dealt with on a case-by-case basis. And now just to go through and look at this as a very broad workflow. I've told you today about our work in reprogramming. And one thing that we need to grapple with, I think, as a field, and we're doing it for sure at Sloan Kettering right now, is this issue of having a naive ground state versus a primed state of pluripotency. Okay, so this was published by Hae-Soo Kim, who's now running her own group at Johns Hopkins. She's running a core facility there. And they published this from the Studer lab in 2011. And what they said in this paper is that iPS cells generally fall over here closer to the ground state of pluripotency overall, whereas embryonic stem cells as a whole fall closer to this prime state. And what that means is that the ES cells are better at neural differentiation than iPS cells. And this was something that we already knew through experience. And what we really would love to be able to do is collapse or change the pluripotency state that these cells are in. Can we add or find ways to try to move them in this spectrum so that, again, they're all at the same place and we're able to compare cells equivalently? when we're doing these sorts of studies. And obviously, there's a number of different ways that people are exploring to try to get that done. One of the things, oh, that's actually E8, isn't it? I got that wrong. One of the things we're going to do when we go back to the lab, as soon as I get off the plane, I think, is we're going to order E8 from, again, Life Technologies. That's just starting to sell that. And we're hoping that by having a simplified medium, that it's going to possibly move these cells in the pluripotency spectrum. But we don't know that for sure. And we're actively exploring lots of different methods to try to move that. And it's possible that we'll have to go back and revisit reprogramming as a way to try to get cells to move where they are in that spectrum. And that's something that's obviously going to be moving a lot, I think, in the very near future. I just told you about my luck in not having to derive a differentiation protocol for this study. But having worked with a lot of different labs, I think that this is something that people will frequently underestimate. And as a field, we've got to get much better and much more precise at how we're able to make different cell types if we're going to ever use this to model disease correctly. And then finally, I told you about different ways that we can go about purifying ourselves at the end here. And I want to end by just saying that this is not the finish line over here, but this is really the start. And it's going to be up to us in each of our labs to be able to find clean phenotypes that we're going to be able to use so that we can understand the molecular underpinnings of these diseases. And so with that, I'd like to end and just say thanks to my lab, a number of the people in my lab that have contributed to the study. Lorenz Studer, who's my boss and a frequent collaborator, Asif Maroof and Stuart Anderson, who contributed, again, to the differentiation. In the autism study, there's Ted Brown and his coworkers at the Institute for Basic Research at Staten Island, and Liz Fernell and Nikos Venizelos in Sweden that have been collaborators. I need to thank the STAR Foundation and NISTEM a lot for giving me the funding to be able to do my work. And of course, I need many thanks to Noemi Fusaki, who I see here in the audience now. Thank you very much. And to Dina Vec as well for being wonderful collaborators over the years. And last but not least, thanks a lot to Life Technologies for giving me this chance to talk. But you know, I wouldn't be here if I didn't believe in them, to be honest with you. And I want to thank David for extending the invitation, but also Elaine Maxwell is really the person that has to deal with me on a frequent basis. So she deserves the most thanks, I think. But she's been able to really give me technical feedback. She's been able to engineer sharing of information that's unpublished between different labs. They've helped me a lot with trying to transition my work to GMP for projects that I didn't talk about today. So I really appreciate the open dialogue that we've had.